What is gene editing? Gene editing, also known as genome editing, is a powerful tool used by scientists to modify an organism's DNA with precision. It allows them to add or remove or change genetic material at specific locations in the organism's genome. Gene editing has become an integral part of biotechnology in our daily lives, particularly in the field of medicine. It transforms the traditional healthcare system and paves the way for molecular medicine to thrive. What led to gene editing and why do we have it? Ever since scientists realized that changes in DNA cause cancer, they have been searching for an easy way to correct those changes by manipulating DNA. Although several methods of gene editing have been developed over the years, none has really fit the bill for a quick, easy, and cheap technology. But a game changer occurred in 2013 when several researchers showed that a gene editing tool called CRISPR could alter the DNA of human cells like a very precise and easy to use pair of scissors. How does gene editing work?
In a document, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, that function is taken on by a system called CRISPR-Cas9. CRISPR is short for clustered regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut-and-paste tool for DNA editing.
Why do we have gene editing and why do we need it? Gene editing has emerged as a transformative technology due to its ability to precisely modify the DNA of living organisms. It provides us with a powerful tool for understanding the function of genes, unraveling the complexities of genetic diseases, and developing potential treatments. Overall, gene editing holds immense potential to improve human health, advance scientific knowledge, and address pressing challenges in various fields. How is gene editing in our daily lives? Treating genetic diseases. Gene editing holds promise for treating genetic diseases by directly modifying the DNA responsible for the condition. Researchers are exploring its potential in treating conditions like sickle cell anemia and Huntington disease and many more. Agriculture and food production. Gene editing can be used to improve crop yield, enhance nutritional content, and increase resistance to pests, diseases, and environmental conditions. For example, scientists are investigating the development of genetically edited crops that require fewer pesticides, are drought resistant, or have increased nutritional value. Disease prevention. Gene editing techniques may be used to modify the DNA of organisms like mosquitoes, potentially reducing their ability to transmit diseases such as malaria, dengue fever, or Zika virus. By altering the genetic makeup of disease factors, it could be possible to limit their ability to spread pathogens to humans. Benefits and risks. Genetic therapies curing, preventing, and treating. In the future, genetic therapies may be used to prevent, treat, or cure certain inherited disorders. They also may be used to treat cancers or infections, including HIV. Genetic therapies that are currently approved by the FDA are available for people who have a rare inherited condition that leads to blindness. The FDA also approved for people who have blood cancers. Recent advances have made genetic therapies much safer. Better safety has resulted in the FDA approving some gene transfer therapies for clinical use in the United States. Personalized medicine. Personalized medicine shows how an individual's genetic makeup influences their response to medications. By editing relevant genes, researchers can gain insights into how specific genetic variations affect drug metabolism, efficiency, and safety. Health issues. Genetic therapies hold promise to treat many diseases, but there are still approaches to treatment and may have risks. Potential risks could include certain types of cancer, allergic reactions, or damage to organs or tissues if an injection is involved. Effects on offspring. Gene editing, like targeting the sperm, eggs, or embryos in humans, would be passed on to the patient's children and could have unanticipated effects. Limitations. The delivery of certain proteins, such as monoclonal antibodies, enzymes, and growth hormones, have been suffering from several obstacles because of their large size. DNA being cut out of the target gene. A major pitfall is that CRISPR sometimes cuts the DNA outside of the target gene, what's known as off-target editing. Scientists are worried that such unintended edits could be harmful and could even turn cells cancerous, as occurred in a 2002 study of gene therapy. An experiment. Treating blood disorders. The approach taken to treat these blood disorders with CRISPR technology in the most advanced trial doesn't directly correct the gene variants that cause disease. It uses a clever workaround. Instead of restoring healthy adult hemoglobin, the goal is to increase levels of fetal hemoglobin. This is a form of hemoglobin that fetuses don't make in the womb, but children and adults don't make. Scientists don't know why yet humans switch from one form of hemoglobin to the other after birth, but fetal hemoglobin is not affected by the sickle cell mutation and can take the place of defective adult hemoglobin in red blood cells. Conclusion. In conclusion, gene editing holds immense promise for advancements in medicine, agriculture, and other fields. However, its unethical implications and potential risks must be carefully considered and regulated. By striking a balance between scientific progress and responsible application, we can navigate the complexities of gene editing and harness its potential for betterment of society.